Good afternoon to everyone. We are PhD students from the University of Girona, and we are going to talk about genetic basis of arrhythmogenic diseases. So first of all, I would like to explain how is electrical transmission in the heart. It begins with an electrical impulse at a particular cells called pacemaker cells, which we can see here in green. Then this electrical impulse is spread through the heart, producing the typical contraction of the atrial and ventricular capacities. Any alteration of the electrical conduction may lead to a lethal arrhythmia and finally could lead to a sudden cardiac death. Sudden cardiac death is the final symptom of different cardiac diseases. The incidence is about 1 per 1,000 individuals per year, and the most common cause is heart attack. However, nearly about 20% of the cases may have genetic origin. It is especially suspicious in young people. So there are two main groups of pathologies which are related to sudden cardiac death. The first one is cardiomyopathies. Cardiomyopathies are characterized by an abnormal structure of the heart. In the upper picture, we can see a normal heart, while on the bottom picture, we can see how the wall of the ventricle is significantly higher.
In some cases of sudden cardiac death, there are a presence of structural dysfunctions in heart. This is the case of channelopathies. The channelopathies are due to a dysfunction of a transmembrane protein that are responsible to pass the current through the cells. This ion, here you could see, at the top you could see a normal heart rhythm. If there are an alteration of one of these proteins called ion channels, this alteration could produce an altered heart rhythm and then could produce a lethal arrhythmia that could end in a sudden cardiac death. As we said, some of the cases of sudden cardiac death are genetics, then heritable. When a patient suffers a sudden cardiac death or is diagnosed in one of these diseases for any symptoms like syncope or arrhythmia, is recommended genetic analysis. Then the aim of the genetic analysis is to find the mutation in one of the related genes, or if we not found the mutation, to search for a new genes. In both cases, our research are focused on found in which mechanisms these mutations produce these lethal arrhythmias and sudden cardiac death. To study these mutations, we produce the mutant proteins, and then we express these mutant proteins in cellular models to see which are the changes produced for these proteins, for these mutated proteins. And now, Mireia will tell you which are the studies that we do for cardiomyopathies.
As we said before, cardiomyopathies are characterized by an abnormal structure of the heart. So we are looking for mutation which potentially can produce an effect in the cells of the heart. So on the left, we have a wild-type protein, as we can see here. As we can see on the left picture, the wild-type protein is well located at the plasma membrane, and cell-cell unions are a particularly important proteins in these cases because cell unions are responsible for the tissue strength. So we are looking for mutation in proteins which take place in cell-cell unions. On the right picture, we can see how mutated protein is not located at the plasma membrane, and it is completely retained at the cytoplasm. Other approach to study cardiomyopathies is to perform histological assays directly on the human heart tissue. Obviously, we only can do this in postmortem samples. On the left, we have a normal heart tissue, myocardium, and we have stained in orange the cells of the muscle heart. On the right, we have also in orange myocytes, but we can see how these myocytes are clearly replaced by fatty tissue that we can see in white. So now Helena is going to explain how we study channelopathies.
As we said, channelopathies is a current defect. Thus, we have to measure the alteration in the current for the mutated protein. In order to do that, we did a technique that it's called patch clamp. We patch the membrane of the cell and we measure the current that passes across the cell. Here we could see the mutant, the wild type current, and an action potential also from a wild type cell. But if we compare with the mutant protein, we could see an alteration in the current and the action potential that we could extrapolate that probably this mutation could produce the lethal arrhythmia in our patient and then sudden cardiac death. Finally, we want that you take home 4 messages. One is that sudden cardiac death may have genetic origin and it's especially suspicious in young. Sometimes sudden cardiac death is without previously disease identification. It's the first symptom that you could manifestate. Another thing is that cardiomyopathies as sudden cardiac death related diseases associated with anatomical alterations. In contrast, channelopathies are electrical inherited diseases but without an alteration in the structure of the heart. And finally, our research and our future work contributes to better understand the mechanisms underlying sudden cardiac death. Thank you for your attention and thank you organizers for JIPI.